nanoHUB-U, online instruction.
Welcome back. I'm Professor Rickus. In this lecture, we're going to continue our discussion on cells as sensors. So we'll talk about sensitivity and in particular look at sensitivity amplification in cellular systems and specifically focus in on some of the design principles cells use to achieve ultrasensitivity. So let's remind ourselves about what a sensor, or in this case a biosensor, is, right? And we talked about wanting to detect some target entity, a molecule, an ion. It could be any sort of biological signal with a recognition element, then some component that transduces this into a measurable event that's measured by some detector and then reported to some output end user. So let's look at a little bit simplified version of this. And in this view, a sensor, if we lump in all of these things from recognition and transduction and detection, a sensor ultimately takes some signal input and converts it into some output response. And that's what we're going to be looking at, how cells do this and converting this signal input into an output response. So let's define what sensitivity is. Now, sensitivity, if we think about what the response is as a function of the input signal, S, the sensitivity is just the differential change in R as a ratio to the differential change in S, right? So in this case, in the simplest, if we were to just plot the magnitude of the response as a function of the signal, and if this were a linear response in the case we have here, the sensitivity would just be the slope of our response curve. So in looking at this, if we were to say a system, a sensor, were more sensitive versus another system, that would simply mean in this linear response curve that the slope of our response as a function of signal would be greater for a system, a sensor that is more sensitive compared to a sensor that is less sensitive. And in this case, if we were to fit our response to a line, the slope would be our sensitivity, and our intercept would be the background signal from our system. Some sensors may have some background response, even at a zero input. So in most cases, our calibration curve, if you will, of the response versus signal is often actually more sigmoidal, right? Because we have some limits at the minimum and maximum. And this defines some range that we'll call our dynamic range. So this would be the level or concentration range of our signal for which the response is linear or predictable, to be more general upon the signal level, right? And so in our biological systems, this saturation out here of our response curve is usually from some saturating of the physical saturation of the response, such as binding. In the case of two molecules binding, there's only so many possible binding sites. So in that case, increasing signal once you've saturated the binding sites doesn't further enhance a response. And similarly, down in the minimum level, we usually are limited by our signal of our response getting lost in the noise. And this would be defined, every sensor has some limit of detection. What's the smallest size of signal that it can measure? Okay. So we've gotten through some definitions here, and we talked about increasing the sensitivity of the system. And in the classic example, right, biologists have known that sensitivity amplification has existed in cells for many decades, right? And the classic example of sensitivity amplification, particularly magnitude sensitivity amplification, which the example we just looked at, is a case that we've already talked about in one of our previous lectures, and that's phototransduction. If you remember from our case study of the photoreceptor, we talked about one photon absorbing event in rhodopsin being translated into the hydrolysis of 10 to the 5 cyclic GMPs in this signaling cascade. So this is one of the classic examples of magnitude amplification here. I've got the light in our rhodopsin event as our signal, right? In this case, looking at the response in terms of our number of cyclic GMPs, and we have this gain on the system. So let's think about how we can engineer a cell to be a sensor. Now, and the most common and simplest way that this is done relates back to some of the lectures that we talked about in looking at gene expression. So a cell can act as a sensor if the transcription of some protein, right, some gene that have some protein product that we can use as a response, okay? And in a way that it is controlled or regulated by some signal, which can be a transcription factor, as we talked about in our previous lectures, to control either up or down the regulation of the production of this response gene. Okay, so the most common, there have been quite a number now of engineered proteins. The most common way to do this is to use either a fluorescent protein, such as green fluorescent protein, and now there are an array of all sorts of colors of responder proteins that we can use, or another enzymatic production of light using bioluminescence. So by producing some protein that we can monitor via light, either emission from fluorescence or light production from photoluminescence, we have a response signal that we can measure as a function of some signal. So this is really our simplest gene circuit that we looked at. Simple gene activation or repression can be used to make a whole cell biosensor, as we discussed in previous lectures looking at gene circuits and simple models of gene expression. So now we're just viewing that same thing and constructing it as a sensor that is useful to us. So let's look a little bit more closely at this. So now if we connect our whole cell biosensor and model this as simple activation as we did previously, right? And we can look at the response of our sensor. Now if you recall our simple activation model, right, where we had the rate of change of the production of our, in this case, response molecule. So this would be our green fluorescent protein or whatever fluorescent molecule or bioluminescent protein that we choose. That would be a function of now our transcription factor we're looking at as our signal, or X, right? So now we can look at, if we think about the production rate of our response gene, our GFP, our fluorescent molecule, as our response, and we plot the transcription rate of that response gene, Y, as a function of our signal, S, we get our familiar Hill function type sigmoidal curve here, and this beta naught now represents, many times we have some background transcription in our systems. So this would be some background signal level, right? And this now would represent our calibration curve, looking at the rate as a function of signal for our sensor. So if our, in our Hill function, our Hill coefficient is just 1, right? We have basically our classic hyperbolic response as our calibration curve. So something we're going to do here now is to normalize the response and the stimulus signal. We're going to normalize our response signal relative to the maximum possible signal. So here now in our Hill function, I've just subbed in our response, right, for some maximum response transcription rate. So this would be what our actual production rate is, and this would be what our maximum is. And I've just subbed in for response and signal into our Hill function here. And we're going to normalize our signal level relative to our, what we were previously calling K. Now in our context of sensors, we're going to call this the signal 0.5. This is the concentration of signal where the response is at half maximum. And we're going to define these dimensionless responses now, little r and little s, simply by normalizing r to r max and s, our signal, to s sub 0.5. And we're going to sub in now, and this will simplify now our equations to our dimensionless form, where our response equals our signal over signal plus 1. So this is our dimensionless form of our Hill function with an n equals 1. And now we can take the derivative and get our dr ds, right, to give us a sense of our sensitivity of the system. And this equals 1 over s plus 1 squared. So if we want to amplify our sensitivity now, let's define a sensitivity amplification factor. Now we're going to look at sensitivity amplification as opposed to magnitude amplification. And sensitivity amplification, we're going to look at an increase in the percent change of response R relative to the percent change in our stimulus S. And we can define now this sensitivity amplification factor, A sub S. And you can see here now we are going to look at simply the change relative to the initial, and again, over the change in signal relative to the initial. So we can rearrange this around to get a simplified form of our sensitivity amplification factor. Okay, so let's look at a curve now of our normalized, so little r, which equals r over our maximum response. So this is our normalized response versus our relative stimulus. So this is our normalized signal, right? Looking at our signal relative to the signal at half maximum response. And so our hyperbolic sensitivity, this is our classic hyperbolic calibration that we just looked at. This is our Hill function, essentially, with an n equals 1, right? And if we look at this response now, and we compare to, we can also generate systems that we'll define as either subsensitive or ultrasensitive. And if we look at this shift, particularly in this range, if we look at this range between 10 and 90% of the response size. So this is this width of our stimulus that gives us between this 10 and 90% range of our total response. And we look at the steepness of this curve. If we have a subsensitive system, and we're looking at the slope here now of response rate versus stimulus, we can see we get steeper. An ultrasensitive system would have a steepness that's greater than our hyperbolic sensitivity. And a subsensitive system would have a slope, right, it's less steep relative to our hyperbolic sensitivity. And for a subsensitive system, our amplification factor, right, our sensitivity amplification is never greater than 1. It's always subsensitive for any range of signal that we're looking at. But for an ultrasensitive system, we get some amplification, right? A sub S is greater than 1, not for all stimulus levels, but for a range. So in this range right in here, you can see it's very, very steep. Now, the other thing to note from this curve is that there is a trade-off between sensitivity and dynamic range. So if we look at the ultrasensitive curve, right, we get that steepness, but the range of signal in which our system, our sensor, is sensitive to our signal, that range has gotten much smaller. And that's highlighted here if you look at A, which is our ultrasensitive system. This is the signal range in which our response is between 10 and 90% of the maximum. You can see it's very tight. So in this signal range, the system is very sensitive, right? But outside of those ranges, we don't get much change in response, and it is not sensitive at all. So there's this trade-off between dynamic range and sensitivity, right? So the ultrasensitive system has a much more narrow signal range in which it responds compared to hyperbolic or sub-sensitive systems. Okay, so now let's talk about, now that we've defined what ultrasensitivity is, let's look at some biological examples and particularly look at some of the biological design approaches that can be used to achieve ultrasensitivities. Now, these occur in natural biological systems, and we can use the same strategy to engineer systems when we want to achieve ultrasensitivity in a sensor system. And so we're going to look at cooperativity, which we've looked at before previously in our gene expression models. This comes down basically to, for example, the increasing the N on our Hill function. We're going to look at cascades, and we're going to look at something called zero order ultrasensitivity. So this is to remind you, this slide should look familiar. This slide comes from our lecture on simple models of gene expression. And if you recall here now, we're looking at our transcription rate of our gene. So this would be just equivalent to our whole cell biosensor example we just looked at. So this could be the production rate of our response signal, our green fluorescent protein, or whatever our response molecule is, as a function of our transcription factor, in this case, our signal that's modulating the expression of our response. And if you recall, right, when we increase the N on our Hill function for gene activation, this response curve gets steeper. So this N equals 1 is our hyperbolic sensitivity. And as we increase N, this gets steeper. The range in which we're sensitive gets more narrow, right? And it becomes more obvious as N gets greater. Now remember, however, there is some limit on this because it's very rare that via cooperativity, our Hill function coefficient is very much greater than 4, right? 4 is usually 4 or 5 maybe is about the limit. And remember, in our gene expression model, this really came from one of the primary ways was through stoichiometry of our transcription factors as they were binding. So we can get some steepness in ultrasensitivity, but there is some limit in how ultrasensitive we can get with this approach. So there are other ways. Another way to design ultrasensitivity into a system, or a way that cells natively achieve ultrasensitivity, is through multi-stage cascades. So what you're looking here is some work where they built synthetic circuits. So this first, our circuit 1 that they built, is a simple circuit where they've taken now the expression of the first gene, the TET-R, which is a repressor of the next gene, which produces, in this case, EYFP, right? So another fluorescent protein here as the output response. And the circuit is kicked off. Our signal here now is our ATC molecule, and our response would be our fluorescent protein. Okay. Now they compared this simple one-step circuit to a cascade of 3 steps. Now in this case, we have a cascade of 3 repressors. So now, and remember, 3 repressors in a row is going to be the same as 1 repressor. So here we've got the expression of our TET-R, which represses the next production of our LAC-I, which represses the next expression of our CI, ultimately to repress the EYFP. So now we've got this circuit, again, of signal input, right, our ATC, and our response molecule, this EYFP, our fluorescent protein. So now what they did was to compare the response, the output-input, the response signal curves. So this is the calibration curve, just like we've been looking at, of the response. In this case, we're looking at the mean fluorescence, which will be related directly to the number of EYFP molecules. And so we're looking at a steady state response here now. And if we look at this and compare the response curve for circuit 1, you can see something that looks a bit more like our hyperbolic. And if you compare this now with circuit 3, the dark diamonds, you can see that in their synthetic cascade, they achieved ultra sensitivity. It is steeper than the simple circuit here now. Okay, let's talk about yet another way to achieve ultrasensitivity. And this is one that is very common in cellular systems. So we're going to take a step away from genetic circuits right now and think about protein circuits. Okay, so in this case, we've got some response molecule. So imagine that this W molecule is a protein that can exist in two states that we'll just simply call W and W star. And we've got two enzymes, enzyme 1 and enzyme 2, that catalyze and control the switching back and forth between converting W into W star and W star back into W, right? So a common example of this might be phosphorylation, where protein W is phosphorylated, right? And gets unphosphorylated. So now, if we have our signal S is an activator of one of these enzymes, we construct this circuit now and have this response looking at the basically mole fraction of how much of our protein is in the state W star will depend on how much of our signal is around. Okay, so let's look a little bit more closely at this, and we'll assume that our enzymes follow sort of classic enzymatic kinetics, Michaelis-Menten kinetics, which is very similar in structure to our Hill functions. Okay, so this structure is very widely used by biology. In intracellular signaling cascades. So I mentioned before that one very common way, that example of this might be phosphorylation of our protein. And so here now, our E1 would be a kinase that phosphorylates our protein. Our phosphatase would be an enzyme that removes our phosphate group on our protein. Right, and in this case, our signal would activate or modulate our kinase activity. And in this way, how much of the protein is in the phosphorylated state would depend on how much signal we have around. So as I mentioned, the structure is very widely used. Here's some estimates. It's estimated that about 2% of the proteins in most genomes are actually kinases. There's some examples and estimates from E. coli and yeast, and you can see across all of our major model organisms, as well as us, we have quite a few important kinases, and kinases are often important targets, drug targets in signaling systems, including things like cancer. So, one thing about this, why do we call it zero-order ultrasensitivity? So zero-order ultrasensitivity depends on some certain conditions for our enzymes. And that is that these will often operate in the zero-order regime. That is the total amount of our protein, our response signal protein, that we'll call WT, which is a sum of the amount of protein in the two states, must be very high relative to the KMs, the Michaelis-Menten constants, of the reactions catalyzed by REM. And so if we look at this, so it turns out the extreme of ultrasensitivity that you achieve depends on this ratio. So if we have a Km to response protein ratio of about 0.1, this is about equivalent to having a Hill coefficient of 2.9. But if we alter that by another order of magnitude in extreme, you can see we hit an equivalent Hill function coefficient of 13, which is much higher than we would be able to achieve through cooperativity, right, and stoichiometry. So we can also, and cells do this, combine cascades with zero order ultrasensitivity to further enhance ultrasensitivity. So this is some model output this is work from Goldbetter and Koshland, names that you'll see very much associated. Were pioneers in a lot of understanding ultrasensitivity and did a lot of classic work in this area. And this comes from one of their papers in 1981, looking at a cascade now of these zero order sensitivity systems. And you can see when we add a second cascade, so now our phosphorylated protein actually modulates or affects the enzymatic activity in our next cascade. So our response becomes the signal to the second circuit. And so now we have two responses that we can look at ultimately as a function of our initial signal S. So if we look at the mole fraction of how much W star, say phosphorylated version of our protein, and how much we get of Z star, or phosphorylated form, for example, of the second protein in the cascade. And now we look at the response curve. So this is again the mole fraction or response of our signal for W star and Z as a function of our normalized signal level or little s. We can see the first cascade I've pointed out here in green for W star. We can see the ultrasensitivity. Now compare this with our second cascade, that gets even steeper, and we get very near like switch-like behavior, right, in this extreme. And that's what happens as our ultrasensitivity becomes stronger and stronger. Our system begins to behave more like a switch, that there's a wide range of concentration levels for our signal in which the response is very low, and then another large signal level where the response is very high. And so we can switch back and forth between these two states of being nearly non-phosphorylated or nearly all phosphorylated. Okay? And if we look at what the equivalent to this would be, if we were to fit this curve to a Hill function for our W star, our first cycle, this would be equivalent to a Hill coefficient of 3.6. But by adding the second cascade in the second cycle, this steepness, if we were to fit to a Hill function, would give us a Hill coefficient, an N of 7.5. So you can see we've nearly doubled the Hill coefficient equivalent from the first cascade to the second in our system. So we started talking about the switch-like behavior as the ultrasensitivity becomes more extreme. And from an engineering design point of view, the analogy to the transistor has often been made. So as we view these switch circuits, either in proteins or at the genetic level, when we start to view them as sensors, amplifiers, and switches, we can apply the abstraction and begin to think about them and putting them together in the same way that we might transistors for electronic systems. And this is often useful in our hierarchy of an abstraction of thinking as we think about engineering design and apply that to how we design and use the protein and genetic circuits that we've been talking about to achieve things like this switch-like behavior and ultrasensitivity and sensitivity amplification. So with that, we'll wrap up some of our device work. And coming up, we're going to move on to bigger picture thinking about future technologies, what synthetic life is and might be, And with that comes the necessity to start thinking about ethics and science and society as we think about some of the future things that we might build using our new skills in cellular design principles. So I hope to see you next time.